Hey everyone, thank you so much for joining us on the joint webinar between Genomenon and Illumina earlier this morning. Or if you hadn't joined, I hope you enjoy the recording of the panel discussion. I just wanted to tag on to that webinar recording because there were a couple of questions that we weren't able to get to in the Q&A portion. There was a great deal of discussion. The panelists had a number of great points to make. And so I definitely didn't want to neglect to bring up a couple of questions that came up. So I'll read them and provide my best answer. Here we go. How to identify biomarkers in the preclinical setting for clinical trials regarding the right model and strategy? It's a question from one of our audience members. And actually that's a question that's fairly appropriate for me to tackle. And in my work with the panelists, we've addressed some of these issues through some of the offerings that Genomenon provides. So specifically as pertains to the preclinical setting and understanding the right model and the right strategy for either your trial design, your companion diagnostic, or otherwise your testing strategy if we're truly talking about a fully preclinical R&D effort. And so we heard from our panelists a little bit about the genomic landscapes that Genomenon provides. And this question allows me to provide a more full-bodied answer to what that looks like. So effectively, as I said at the beginning, Genomenon is organizing the world's genetic and genomic information. We are gathering genes and genetic variants and the mentions in the context of diseases and phenotypes and drugs across many decades of scientific publication, both clinical studies and functional studies. And we ally that with information in the public domain, such as population frequency, as well as predictive models of pathogenicity. And together, for any given gene, all of that information for every genetic variant ever described in those references comprises a genomic landscape. So I think you heard each one of the three panelists from the pharmaceutical companies allude to the work that we had done with them to produce some of these genomic landscapes. And so in the preclinical setting, that comprehensive view of all of the genetic variants and understanding all of the functional studies and all of the models, systems, and assays that are used to design, to divine the functional consequence of those variants, that is patterned in the comprehensive genomic landscape that we can provide. So if any of you in audience has any such need for preclinical information coming from those genomic landscapes, we'd be happy to have a conversation. The second and final question that I wanted to be sure to get to share with the entire audience goes, well, it is clear that comprehensive genomic profiling via next generation sequencing, CGP via NGS, is the new standard. It would be nice to hear comments from the panelists, my apologies for not being able to get to this question, on the need to improve accuracy and earlier detection of actionable mutations to inform therapy selection sooner. So I agree that would have been a great question to address with the panelists. I had it on my legal notepad. We just didn't have enough time to get to it. However, I'll take a stab at it and emphasize that it comes in the context of circulating tumor DNA, where the sensitivity is paramount, because if you're trying to monitor disease and detect low levels of disease, if you don't have a highly sensitive assay, a negative result may in fact obfuscate the idea that the patient is actually still with disease. And similarly, the specificity of those results, which is a question that I wanted to address in the context of the comprehensive genomic profiling, has to be high, has to be very high, so that you don't run into a false positive over-treatment situation. And so we didn't touch on sensitivity, specificity by name, but we did entertain some of those concepts in the questions and the answers that the panelists provided. So I don't know if this question came earlier or later, but I definitely wanted to bring that up to share with the other audience members, to say that particularly when we're looking at circulating tumor DNA, the sensitivity of the assay from blood draw all the way to interpretation, as well as the specificity of those results and the interpretation of their clinical meaningfulness will definitely be an important consideration. So thank you to whoever asked that question. It's quite likely that we'll be reaching out to you directly. And I'll conclude by saying what I said at the end of the webinar. If anyone has any questions that would be best addressed by myself about Genomenon and the comprehensive genomic landscapes, or otherwise Dr. Eberhard and the comprehensive genomic profiling that Illumina offers, please don't hesitate to reach out to us. Thank you so much.